We are looking at a very small slice of liver tissue, which has cancer cells. And so Demir is a researcher at QI Tissue, works in partnership with University of Oregon, and we built this environment to basically collaborate around this analysis.
Hello, welcome to this virtual reality presentation. So what you see in front of you is a piece of tissue. It's a fairly long, narrow strip, which is typically collected using a needle. So it's called a fine needle biopsy. In this fine needle biopsy, you see the nuclei of the individual cells colored with little bluish blobs. So what you see primarily for the cells that are not colored with any other color, they are just showing the cell nucleus. Surrounding it, for some of the cells, you'll see the cell body, which is colored in some cases with a green dye, an indicator of tumor cells. So from the point of view of a cancer researcher, one of the big problems with these highly multiplex image data sets, like the one we're looking at here, the size of the image is enormous, the number of channels, and so the number of colors that you would want to see is enormous. So in order to interactively work with this data and see the spatial relationships, which is what imaging brings you, between the individual biomarkers, is what a VR environment is particularly good at, because I can be in the environment itself, look at the individual cell clusters, the spatial relationships.
So think about an 8 gigabyte image, thousands of pixels with several dozen different layers of possible biomarkers that help describe exactly what you're seeing in that image. And what we're doing here is I'm actually turning on and off different levels of different biomarkers, so you can understand what's going on with this particular sample.
In that area, what we'll see are a number of reddish streaks. The reddish streaks are blood vessels that are labeled. So here we see in red these blood vessels that are sort of on the periphery of the area with the tumor cells, but also we see quite a few of them in the area of the tumor cells. That is an indicator that the tumor cells are recruiting blood vessels to grow, or they're actually stimulating blood vessels to grow in order to feed the tumor. So the tumor is actually trying to feed itself. Clearly something that is not very good for the tumor going away. So far we've been looking at the biomarker view, one of the two views that is available within this environment. What we really would like to know is, what are these cells? So we want to identify the cells. So we can use the biomarkers using machine learning based on their biomarker expression status of all the biomarkers. We then can classify them into classes. Now each of the cells is colorized in a specific way. The nice thing now is that in order to figure out exactly what is where, because this is still a very complex scene to look at, we can just turn on and off the individual cell classification markers. So let's turn off a few. I'll turn off the stroma, and you just saw way in the back there. I'll turn it on again.
So one feature that we've built here is to be able to overlay additional feature data on top of the cell data at any point in time. So in this case, in the cell classification view, if I turn on the tumor cells, I can have these, lollipops, these dots and lines, which give us a sense of the strength of the expression of that particular biomarker. And each one it's actually tied to the, whatever cell classifications I have turned on here. So now I can see the higher expression of an immune cell or a lower expression of an immune cell based on the height of the bar. And then finally, on the far side over here, we also have additional scatter plot of the dots, but actually each and every one of these axes can be swapped for another valuable informational visualization. So taking a look at different cell states, etc. So the researcher can combine 3 different axes, 3 different properties together to see relationships between them.
All of this is interactive, and that all participants can basically choose from a different selection set of options to really analyze this really amazing thing to see come together here, and see the power of building these immersive environments to collaborate around.